I guess I will start from beginning. The purpose of this is to examine further the data for the example that Chad presented 2 weeks ago about the distribution of ERBB3 gene and some artifacts that he uncovered. And I will talk a little bit about what we found out, and then I'm going to talk more about the assay itself and the low-level processing of this data that may be relevant in this example. And I will also suggest an internal benchmarking strategy for whole signatures that was also suggested by Arvind in kind of a conversation, informal conversation, but he believes that that's the best way to perform first pass benchmarking of any signatures that we create. And I'm going to talk a little bit more what exactly this means. So hopefully I'll be done within 20 minutes, and please interrupt me with questions if something doesn't make sense, or if I'm done quickly, then maybe somebody else can chime in with anything regarding this problem. So the first thing we did was to recreate Chad's results. So what Chad did, he looked at the distribution of ERBB3 gene in A375 cell line, and he looked at an empty and untreated profiles, assuming that the distribution should be fairly simple across all these profiles for this one gene. And he looked at level 3 data. Turned out that there was this artifact, that there was a significant portion of genes that had very different gene expression level. And the first thing we did, we recreated his results just to make sure that we are using the same data and talking about the same gene. And as you saw on the left-hand side are Chad's figure, on the right-hand side are our figures. They are the same. The lower one looks a little different because we are plotting densities and not counts. So the next thing that we did is actually look at level 4 data. And again, level 4 data, these ZMAD scores, and this is the level that Broad Institute advocates for people to use. If you were to use this data, they will tell you that you should probably start with level 4 data because this is the final massaged, normalized product that should perform well in the setting that they think most people will be using these signatures. And again, that's not necessarily the case that all of you want to use it in the same way. But this is the final product for this data. And as you can see, so on the right-hand side, on the left-hand side are still the original figures for level 4 data, and on the right-hand side are the equivalent figures for level, so the left-hand side is level 3, right-hand side is level 4 data. And as you can see, this bump disappears in level 4 data, so it's normalized out somehow. So the z-scores of ERBB3 across all these same profiles look the way you would expect them to look, meaning that they all seem to be part, or vast majority, part of the same bell-shaped distribution. If we'll split by empty and untreated perturbation description, we still get about the same picture. So these bell-shaped distribution for the two subpopulations still seem to be behaving the way you would want them to behave. So they are normalized properly. And the same is the case if you split further by bead batches that also Chad did. Again, they all fit under one bell-shaped umbrella, and they all are behaving the way you would expect them to behave. So it seems that whatever processing has been done from level 3 to level 4 data has accounted for these different expression levels in the subset of these profiles. So the question is, what happened there? And one thing that I wasn't aware of is that actually level 3 data is not quantile normalized. So if we look at whole profiles, these 2,000 profiles, not just ERBB3, but all 22,000 genes, so measured genes and genes that were imputed. And if you look at their overall distributions, densities, they are actually quite different. So if they were quantile normalized, you would expect basically to see one line here. But it turns out that level 3 data is not quantile normalized. So theoretically, this could account for subset of this measurements for ERBB3 to behave differently, right? If for example, the distribution, overall distribution of these measurements for a subset of this profile is shifted significantly from the majority. So that was the first thing we checked. If we perform quantile normalization of these 2,000 profiles and repeat these figures for ERBB3, you may say that there are slight improvements, but the overall picture remains the same. We still have this bump, and still it's mostly explained by this subset of untreated and bead batch 7 profiles. So quantile normalizing these profiles didn't really remove this artifact. So that's the first take-home message. So there is something else that is going on there. And that brings us back to how does this Luminex assay really work. And I will go through this because I wasn't sure what Arvind was going to talk about last time, but he did not really go into these details. And I think for some of you at least, these may be useful. It may help you with interpreting the data. So the way this, and this is kind of statistician version of this assay, right? So if you want details, there is a paper that actually describes in detail this assay at this level. So the assay is actually designed originally to measure 500 genes. And the way it does it, there are 500 different types of beads, or beads with 500 different colors that can be resolved by the instrument. And beads, these are like very small, tiny beads, and they're coated with the probe. So each probe, a bead with a specific color, is coated with a probe that can recognize a specific transcript. So in our case, this blue bead is coated with transcript probes that can recognize gene 1 transcript, and the green ones are coated with probes that can recognize gene 2 transcript. So what happens is then a typical hybridization assay, that this pool of these 500, lots of these beads, 500 different kinds of them, is thrown into the soup, and then depending on the relative abundance of different transcripts, which are labeled, I guess, or it doesn't really matter. We can think about them as labeled transcripts. So the appropriate one will be hybridizing, and they're going to hybridize. The level of hybridization will depend on the relative abundance of this transcript, just like, for example, in microarray. You can think of this as a spot on a microarray. And then each of the beads, or a random sample of these, you can think of, will be actually identified by the instrument, and intensity level of each bead will be measured. So what's going to happen is that uneven number of probes or beads of different kinds will be identified for actual measurement. So the output data from a single experiment looks something like that in this table. You have a bead or the analyte identifier. In my case, I have two. It's blue and green. In fact, we have 500 of these. And then we have measurement for each of them. So each of these beads will go through the instrument. Instrument will identify its color and record it, and then it's going to measure its fluorescence, and that's going to be the measurement of the gene expression level. So in this case, we have two beads that showed up that were green, one bead showed up that were blue, and so on. So there are many, many measurements for each sample of these. So finally, the summary measurement for each transcript is constructed by some kind of average of all beads of the color, of the certain color, or all beads that were coded with the probe for this particular transcript. So that's how the assay works if we had only 500 genes to measure. So L1000 actually goes a step further, and they need to measure 1,000 genes. So, but still use only 500 different beads. So the way they do that is by using each bead color twice. So in this case now I still have blue bead, right? And this one is coded with the probes for gene 1. But then I have more blue beads that are now coded with a probe for gene 2. And the way that these are going to be resolved is by basically mixing different abundances of the same bead, but coded with different probes. So in particular, in this case, they mix, for example, twice as many beads of the same color that are of blue that are coded by probe for gene 2 than for the gene 1, right? So now it's not only that the information about the fluorescence intensity of each of these beads is important, but also the counts are important because that's going to help us resolve which gene was actually measured, because two genes we cannot recognize them just by looking at the color of the bead. So the way this is done is, if we look now for all the observations or measurements for a particular bead, for this blue bead, we can, assuming that these two genes have differential expression level in this particular sample, we can then resolve two different populations of measurements, right? And because this one population has twice as many measurements as the other population, we would be able to basically assign measurements from this population to gene 2, because it has twice as many beads. We know that we threw in twice as many beads for gene 2 as we did for gene 1, and then these would be assigned for the gene 1. And now the summary is for the expression level of the gene 2 would be some kind of average of these observations in this cluster, and for gene 1 would be some kind of average of the observation of this cluster. So basically a simple cluster analysis then deconvolutes measurements for gene 1 and gene 2 from the same type of bead.
Mario, what is on the x-axis on this graph?
On the x-axis are the measurements log2 scale fluorescence intensities measured on this bead of one specific color.
Intensity, thank you.
Right. so basically we use both intensities and the presumed counts of these beads in different clusters of measurements to resolve which ones correspond to gene 1 and which one corresponds to gene 2. Right?
So can I just ask you, would it be confounded if there was a dramatic difference in the expression level of those two genes? If, say, the expression level of the gene for which there are twice as many beads was significantly lower, wouldn't that cause an overlapping distribution?
Right. So that is a problem. So if this distribution, so in my example here, they're beautifully separated. If for whatever reason that is not the case, that will potentially introduce problems. And again, I'm actually, this is one of the examples that is coming up about the problems. So you're absolutely right. I think that is one potential problem there. So here are the problems, and these are now actually, this is real data. These are documented problems. So this is a pair of genes in one set of observations that we looked at. And so we have gene 1 and gene 2, and they are resolved, and I don't really know which one was more abundant, but that doesn't matter here. But the distribution of actually, so this is this pair of genes across a whole bunch of different profiles, and it turns out that their expression levels seem to be systematically different, quite different. So they're pretty well separated, right? But then there are these small subpopulations of measurements that look conspicuously like the opposing gene expression level distribution. And you can, just by looking at in this view, you cannot really say if these are really outliers or points in which this particular, say, gene 1 is more expressed than at its baseline level. And same thing for the gene 2. But what is conspicuous that basically we have about the same proportion of each of them for gene 1 behaving like the most population for gene 2 and other way around. So we don't have a little bump here for less than normal expression of gene 2 or a little bump here for higher than expected or normal expression level of gene 1. But then if you look at them in this scatter plot, then it becomes even more obvious, because it turns out that these outlying observations for the two genes were measured on the same bead. So not only they behave like gene 1 here and gene 2 for the other one, but they're observed on the same bead. So a simple flipping event in which we accidentally called or mislabeled gene 1 and gene 2 in this deconvolution process would cause this exact data behavior. And that kind of gives us likelihood for this happening by chance or probability of this happening by chance seems pretty low. And this is most likely, at least we concluded at that point, the problem with the convolution. So although overall these genes separate pretty well, because you repeat this cluster analysis many times, and the sampling of the beads that were actually put in this introduced in the system varies, I guess, and this random noise will occasionally result in mislabeling of the two genes. The problem with this particular kind of errors is that they are really big, right? So if you look at, this is change of 7 to 8.5, so this is 2 to 1.5 fold change. So this is significant change over the typical distribution, say, of gene 2, and this can introduce a lot of spurious correlations. I guess that's the idea, if you're not careful. So the other problem, and again, that's something that was just mentioned, is that these expression levels are overlapping, and this may not be such a problem if they're pretty much identical, because then they all have about the same expression level, and that's less of a problem, maybe. But if, for example, one of them is noisier than the other one, then there is really no way to deconvolute this properly, and still we may get some biases. So that's another situation. And then what do you do about this? Well, turns out that even if you implement a simple flipping strategy in which you just look at these z-scores and see whether there is a chance that this flipping event occurred, in which basically the identities of the two genes will flip, and then you correct for that. So if you do this systematically across a large subset of these profiles, you can significantly improve this connectivity that Arvin was talking about. That is similarity, the higher similarity between the replicates of the same treatment as opposed to profiles coming from different treatments. So this is the point of this ROC curve, right? So if we just do the original data, we get a certain level of behavior. There is a little bit more information here, but the point is that this flipped data is always better than having the original data where this is not taken care of. All right, so this is the reason why I'm talking about is that going back to this the behavior of this one gene ERBB3 could potentially be a artifact related to this deconvolution. And then turns out that in the processing of the data, going from the level 3 to level 4 data, in addition to constructing these Z-MADs, the Broad Institute or people in Broad Institute first perform additional quantile normalization, and then second, they have an algorithm that does this correction of these potential problems with deconvolution. So the reason why the distribution of these z-scores for level 4 data behaves much more like you would expect them to behave than the level 3 data may be that after accounting for this kind of events, these artifacts gets normalized out.
Just a quick question about that.
Yes.
Are the z-scores calculated per plate? Because if so, that's going to remove any shift completely, and you're only looking at, and I mean, the z-score is supposed to account for the different levels of variability as well. So is that supposed to be a good thing in this case, or are they actually doing the flipping first and then the z-scores?
You know, I would, I'm not quite sure at which point the flipping occurs, but the way we did it when we experimented with this like 2 years ago was to do it on z-scores, with z-scores, yeah, and they're calculated per plate. And you're right, some of so these events would also affect the variance within the plate for a specific gene, and would affect, because you use this in calculation of z-score, it would affect it in that way as well. But again, the z-score, the way it's calculated in a robust way. So it's actually the average is calculated using medians, and variance is calculated using this MAD estimator. So that and these events are, if they're really high impact events, are probably rather rare, and they would not actually affect construction of the z-score. So again, it's somewhat of a evolving story for me as well. And if somebody has more information on this, please chime in. But I believe that the flipping is done at the end, once you process data, get these scores, and then the flipping correction is done.
I just think that, for example, if you have a whole plate where the gene expression for a particular gene is lower, once you do the z-scores, it's going to be centered at 0, regardless of what the original distribution was. And so I don't see how you can, if you can even see the fact that it needs flipping at that point.
I was planning on sorting this 3 gigs. I have 5, right? I was going to do 2.
Right. If that is the case, that it goes for the whole plate, the expression level for the gene is low, then I mean, it's going to center it out. But so the question that I have, let me go and look, if maybe we can focus on this figure just to talk about it. So if so what we have basically are maybe expression levels on some plates that behave like this, right? So if you just subtract, but if this distribution reflects behavior of this gene within each plate as well, right? So this is behavior across different plates. But if also within each plate the gene expression distribution is like that, is like bimodal, then subtracting, constructing z-scores out of that will still result in a bimodal distribution, right?
Correct. But if what you're seeing here is that you have some of the plates have the genes switched and some don't, then you get 2 separate peaks and each one cancels itself out. I don't know that this is true. I haven't looked at the per plate distributions.
Yeah, I would. Right. So you're saying if actually these two peaks, say for this blue subset, right? These are just untreated.
Right.
If these actually come from different plates, and they would actually describe the distribution. So on one set of plates you have one peak, on the other set of plates you have the other peak. Then this would cancel each other. Yeah, I agree with that. So I guess that's another thing that would be good to look at the per plate distribution for these.
Yeah.
I agree totally. So that could be, if that's the case, then just construction of z-scores would take care of it.
Well, my concern would be if you're constructing z-scores and what you're seeing is that secondary peak is actually the paired gene for ERBB3, then you're getting measurements for that other gene mixed in, and you absolutely can't tell the difference at level 4.
Right, but it's unlikely that you would get this flipped systematically per plate. That one whole plate will have all flipped and another whole plate will have it correct, right, for the same gene and for this one subset. you know what I mean? So for example, I forgot how many there were. Too small. It's like 600 profiles. There are 600 measurements in this blue thing. So what you're saying that basically we have twofold problem here, that maybe one of these peak represents one set of plates, but then also we have flipping happening within this set of plates. Right, actually, maybe we have it happening here. Well, probably here, right? Because this one looks different. So that, that seems unlikely to me, but I don't know. That's the whole plate would have things flipped. I think we should probably take a look at this per plate distribution.
Yeah, I'll look more into it and let you know what I find out.
Oh, yeah. Great. Any other comments? Does this story make sense to everybody?
Well, this is Peter. I have a question. So the probes themselves, are they multiple different regions on the RNA that are being probed on the same bead, or how do they correct for, say, degradation of the RNA? So you had differential degradation. I mean, do they have perfect RNA preparations for each sample?
Oh, they don't. I mean, it's as Arvin indicated. This is a very dirty assay. They don't actually extract RNAs. It's kind of they just lyse the cells.
Yeah.
And my guess is that no, I don't think so. I think it's a single probe per transcript. I mean, the probes are being benchmarked and so on. So and they update, I think, their beads, but I would say they have only one probe for each transcript. So again, it's a high throughput assay that tells you one thing, and then the question is how good it is for any given gene versus how good of a signature it gives if you look at all genes that they measure together and try to connect different perturbations that cause the same transcriptional change. And I think these are two different problems that we may try to address, and this may work better for one kind of problem than the other, and also the data that you use may be different for two different kind of problems. So this brings me to, maybe I'll just throw out this benchmarking at you, and then maybe you can tell me what you think about that as well. So the question is, now we have what Arvin calls biological replicates of each perturbation. So these are plates, whole plates that are basically replicated. And their first pass data quality assessment, in which they actually construct the lists of these gold signatures, meaning that they believe they are reproducible and informative, is basically based on the ability of these replicates to recognize each other. And I kind of borrowed his two of his pictures from last week. And so one of the criteria is that each replicate has to have a certain level of correlation, and their cutoff is Spearman correlation of 0.25. But the other criteria is that they also need to be specific, meaning they cannot just correlate to anything. And in their case, I believe they were saying that they need to correlate better than 1% of just kind of random pairs of profiles. And then if that is the case, they are classified as gold, meaning so the signature now consists of a set of replicated profiles that are highly correlated between each other and are specific, meaning that they're correlated more, significantly more with the replicates or with its replicates than with any other random signature. So, but then the question remains: how do you construct? How do you summarize these replicates into a single signature that can be used then to connect different perturbations? Because this is, again, this first problem that that perhaps this data has been designed to solve is if you have two different perturbations or a disease and a perturbation, and you want to see whether there is concordance in transcriptional response, you want to have a signature of transcriptional response after perturbation and then this other signature, and you see just how concordant they are. So the question is, how do you construct these signatures? And the simplest way, and I think that's how some of us or some other people have been using what was done to construct signatures, is to basically average replicates. Right? So if you have 4 replicates that are designated to constitute a gold signature, meaning that they are highly correlated and they're specific, then you can just average expression levels of each gene, of these statistics, depending where you start, from these replicates, and this will now constitute a signature, meaning you will have a 978 dimensional vector of observations that you can correlate with anything else to see whether the perturbation has a similar transcriptional response to this other profile of interest. The question is, obviously there are many, many different ways that this could be done. And Broad Institute in particular is using this weighted average. There are also other ways to do it, like Avi's group has their own way to construct signatures. But the question is, the first question that I think we need to answer is, what is a good benchmark for establishing which signature is working better in this first type of analysis, which is connectivity analysis. So connecting one perturbation to another. And the way that, again, Arvind suggests that this is done is to use replicated signatures that are present in this data. And these replicated signatures come in for various reasons. They decide to perform exact same experiment that they did, that they already performed, but they just repeat it maybe a year later, or 6 months later, or 2 years later. And the reason for replication is basically for the validation of some of the analysis that they performed, but it turns out that there is a significant number of these replicates in the data already. And these are natural gold standards because we know that they should behave exactly the same, and yet they are complete experimental replicates done with a specific time delay, maybe with the different batches of some materials that are being used and so on. So there is a certain level of likelihood that they will just not replicate, and that happens from time to time. So I guess that's what we did for, and this, as a test case, or basically how we can use these replicated signatures for benchmarking. And we benchmarked the very simplest way of constructing signatures. We looked at just the MCF7 cell lines. I shouldn't. Sorry about that. I'm touching this. I should not. And then basically establish what should be a same pair. So same pair or true positive pair of signatures should be something that had the same duration of treatment and same perturbagen. And again, these are all CP signatures, chemical perturbagen signatures in MCF7 cell line. So these are signatures that were basic experiments that were basically replicated. And then false positive pair would be any two signatures that have different perturbations. And again, we repeat the same analysis that Arvind did on technical, or showed as on technical, or called biological replicates of the same experiment. Now we did it for these replicated experiments and established basically how much more the true positives pairs are correlated in comparison to false positive pairs. And if you run a typical ROC curve, it turns out that it's very, very highly precise, I guess, this separation between true positive pairs and false positive pairs. But using this kind of ROC curve, whole ROC curves, doesn't look like it's going to be useful for resolving maybe a better way to construct or compare these signatures instead of just doing averages and using correlation coefficient or measuring similarity or concordance. So on the right hand side is this, it's again still basically the same information as in the ROC curve, but maybe focusing on just a very high specificity range. So we are looking at 90th percentile to 100th percentile of false correlations for false positive pairs, and then look at true positive rates or corresponding to these false positive rates, where basically each on the y-axis we construct again just the proportion of all true positive rates pairs that had correlation higher than the corresponding percentile of false positive rates. And it just seems that maybe there is a little bit more room here for improvement, and we could resolve this, the method that could do a better job than this simple method, or maybe even methods that does worse. So anyway, that's all I have. It's just a kind of suggestion of how to start with using internal benchmarking of these signatures to pinpoint the best way to summarize them and maybe the best similarity measures that can be used if our goal is connectivity analysis.
Could I ask, from a sort of naive point of view, how do you know that the perturbagens are in fact distinct from one another? You might imagine some can have overlapping responses.
You don't really, yeah. I think there is a lot of these false positive pairs are not clean, meaning there are some true positives there for perturbagens that have basically the same mode of action. There will be these, but it seems that is very that their proportions of this pair is very low compared to really false positive pairs, just from looking at this analysis. So I'm with you. I don't think we can say that, but I don't think it muddies the picture significantly if you just use this for benchmarking purposes.
And is the goal to determine which genes are responding in a characteristic way? That's what a signature is. It's that this perturbagen causes this change in this set of changes in gene expression. And that would be then the significant deviation from, you know, the normal distribution, or z-score.
Right. So I guess it depends what, again, now we are getting back to the objective of the analysis. If the objective is just to connect two perturbagens because they have similar expression profile, then just correlating average z-scores across all 987 genes doesn't seem like a terrible first pass analysis. You could do this many different ways. You could first identify most upregulated genes and most downregulated genes and do some kind of connectivity score that was used, for example, in connectivity map, and is still used by Broad Institute to connect them. In which case you would actually have first decide which genes are upregulated, which genes are downregulated, and so on. So I think all of this, but if the objective is just to find, whether pair of perturbation signatures are concordant or not, then we don't necessarily need to go into gene-by-gene analysis. Although it may perform better if we do gene-by-gene analysis, but I guess this is the first step, kind of, in which we just correlate expression levels of all genes. And again, this is not the main problem that some of you are trying to address, because if for example, the problem is assessing the mode of action of different perturbagens, then you actually want to know what specific genes have changed, how they changed, and so on. So it's a different kind of analysis that may require different kind of benchmarks.
Hi, this is Kai from University of Washington. And Mario, thank you for looking into the data artifact issue. This is very helpful. And I'm wondering, since Broad has done the entire plates being replicates to benchmark their data, I'm wondering if they might have done some internal data to benchmark the flipping effect, the deconvolution of the clusters. If it's possible, maybe we could ask if they have asked for more information exactly how they corrected, go from level 3 to level 4, and if they have any internal data like spike in or something else, that more like we could do more benchmarking on our end as well.
I can pass this request or a question, or both, and and see what, and I will let you know what I hear. You are free also to contact Arvind on your own, but I don't want to say, I don't want to ask this question for you, but maybe it would even help if more people ask the same question, then they may provide answers sooner than if just one person.
Okay, I would be happy to email him directly.
Okay, I will ask him as well. So.
Okay, thank you.
Sure.
Hi, Mario. This is Chanan from Avis Lab. I just want to ask you a quick question about the correlation between the pairs. So what's the correlation level between two true positive pairs again versus the correlation between two false positive pairs, and how much is the difference between these two?
That's a good question. Do we have this somewhere close by? I don't remember exactly, but in my head, I think that the distribution of false positive pairs goes up to about 0.2, I think, or 0.3, and this corresponds to about the 20th or 30th percentile of the true positive pairs, and true positive pairs will go high, right? They will go up to 0.8, but I can give you probably, maybe it's the best that I just give you the summary of these.
Yeah, if you give me all the perturbations you choose to compare, I can easily replicate and using our method, and it's easier for us to compare internally between different methods.
Excellent. That was exactly my hope. we will construct basically all the information that you will need to just replicate, in the table that you can just use and run this on your own.
Oh, thank you.
And we'll be happy to share this with anybody else if you would like to benchmark yourself. Okay, any other comments?
Well, thank you for this.
Okay, you're welcome. Hope this was somewhat informative. I will try to organize the next topic soon. Again, if any of you have anything that you would like to share with the group, please let me know, and I hope to see you next week. Okay.
Mario.
Yes?
Yeah, Peter here. So I don't know. I think that I'm not sure what Stephen Britton's and Clarissa's status is. So I guess I'll let you know if they're willing or want to have information to share next time.
Okay.
This is Mark. Peter asked me to present today, and I've only just joined this group like a week ago, and I haven't even seen the data yet, so it seems like it would be good to get a little more on my feet before I present anything. I can certainly talk about what I've been doing on a different data set, but it would be better to be focused, I think, on what this group is interested in.
Right. On the L1000, that's our situation as well, I think. I really want to, and I'm sure that Clarissa and Stephen do as well, take a look at what the L1000 data can tell us about what we're doing and vice versa. So.
Yes, so my hope is also that we'll keep talking about L1000, and again, if anybody would like to present any results, any kind of angle that they have on L1000, that would be great. But I would also like to slowly evolve to some extent by looking into other types of links data and how maybe they can be mixed or non-linked data with L1000 and go in this direction as well. So.
Great, great.
So that would be great topic. I mean, if you could kind of present this angle, that would be great.
Sounds good.
Excellent.
Okay.
Yeah.
Thank you, everybody.
Mario.
All right.
Yep, thank you. Bye-bye.
Bye.
Bye-bye. Thank you.
Are you still there?
I'm still here.
Yeah? I don't see you anymore.